All right, guys, we are going to be focusing on hearing, touch, smell, and taste. Hearing, touch, smell, and taste. So hearing, touch, smell, and taste. Let's start with hearing. Hearing. So parts of the cochlea, when it comes to the ears, that snail-like tube within the ear. Then we have the basilar membrane. So anyway, let me read some notes that I was taking on the side. So sound waves equal vibrations. The eardrum vibrates, bone in the ear, and these bones vibrate. And so vibration makes fluid more, fluid has hair. No. Vibration makes fluid move, and the fluid has hair. Depending on intonation, certain hairs move. So depending on the high pitch, low pitch, certain hairs move, right? So vibration makes fluid move, fluid has hair, sound waves equal vibrations, hence the movement of these sorts of fluid. The eardrum vibrates, bone in the ear, bones vibrate. So with hearing, parts of the cochlea, which is the snail tube-like structure of the ear, the basilar membrane. Basilar membrane is narrow and stiff near the base, thicker and flexible near the apex, which is the center of the cochlea. So thicker and flexible. So basilar membrane is narrow and stiff near the base, thicker and flexible near the apex, the center of the cochlea. High pitches produce more displacement near the base, and low pitches produce more displacement near the apex, which is a tonotopic organization. So the tonotopic organization, hairs like a piano key. So on the basilar membrane, the organ of Corti contains hair cells. The cilia are smaller hairs, and they have tip links. So the tips of these hairs are linked. The tip of the hairs are linked. And when you hear a noise, these links, the hairs, since the hairs move, they break that link and allow electrical signals to go through. So the link breaks, creates electrical signal. The hair cells move, opens K and Ca channels, so potassium and calcium channels, excitatory channels, leads to glutamate release at the auditory nerve, and transduction occurs. So the organ of Corti contains hair cells, the cilia, smaller hairs, tip links, tip of hair, link breaks, creates electrical signal. The hair cells move, opens K and Ca channels, excitatory channels, leads to glutamate release at the auditory nerve, and transduction occurs. Sound is turned into an electrical signal. Fun facts about hair cells. Hair cell damage, noise, infections, genetic diseases, aging. Higher frequencies are harder to hear. With hair cell damage, higher frequencies are harder to hear. Regenerations can occur in birds and some invertebrates. Generally does not occur in humans. So these cells cannot regenerate, not necessarily in humans, but sometimes in birds. More crucial to the movement of birds. So the hearing is more crucial for birds. People can get new cochleas, but it's all or nothing. You have to get a whole cochlea transplant. You can't get a partial cochlea. So auditory pathway to the brain. Out the cochlea via the cochlear nerve. So the auditory pathway to the brain. Out the cochlea via the cochlear nerve. Ipsilateral connections to the medulla. Ipsilateral is the same side. Ipsilateral connections to the medulla, also known as a brainstem, keeps low and high pitches in separate locations in the brain. Approximately 80% of information projects to the opposite side now. From the medulla. So the idea of pitch is crucial. So the reason why we have two ears is to know where sound came from. So from the medulla, two pathways. Pathway 1, the superior olivary complex in the pons. Information from both ears first combines at the medulla. Sound localization helps to determine where a sound came from, left or right side. Sound localization. So pathway number 1 from the medulla. Two pathways. Pathway 1, superior olivary complex in the pons. Information from both ears first combines. Sound localization helps to determine where a sound came from, left or right side. Projects to the inferior colliculi, the tectum. Pathway 2, directly to the inferior colliculi, the tectum. Thalamus, medial geniculate nucleus, aka MGN, is a primary auditory to the primary auditory cortex. So it goes from the medulla, goes to the superior olivary, complex in the pons. Information from both ears first combines in the medulla, helps to determine where a sound came from. Sound localization projects to the inferior colliculi, the tectum, and pathway number 2 directly to the inferior colliculi, the tectum. Well, they're both the same. Projects to the inferior, pathway number 2 directly to the inferior. Okay, so wait. So there's one for the first pathway will go from the medulla straight to the superior olivary complex in the pons, and then information from both ears first combines, helps to determine where a sound came from, projects to the inferior colliculi. So first it'll go straight to the superior olivary complex. It'll go from the pons and straight to the tectum. Pathway number 2 goes directly to the tectum and then to the thalamus, the MGM of the thalamus. MGN, and very at the end, it reaches its primary auditory cortex, the A1. So what happens after it reaches the A1? Like vision, A2, A3, A4, two pathways. The dorsal is where pathway. Remember, we're looking at hearing, right? So the dorsal, the front, dorsal, where pathway. The ventral is the what pathway. Researchers try and relate auditory systems to visual systems. Some similarities have been found, but nothing is certain. Detecting the pitch of a sound. Place theory, hair cell theory, argues that different pitches activate different locations of the basilar membrane. So there's place theory and there's also frequency theory, argues that most hair cells are activated with all sounds. They vibrate or activated at a frequency similar to the pitch of the sound. Thus, a 1,000 hertz tone would cause the neurons to fire at 1,000 times per second. Both seem to be true. Place theory for pitches, 4,000 hertz, which is high. Frequency theory for pitches, 400 hertz, low. Both mechanisms for pitches in between. Organization of the auditory system. Tonotopically organized throughout. Organized by pitch, by tone, just like the cochlea. The auditory system in the pons, thalamus, and A1 are all organized by pitch. So detecting where a sound is, where, location, that has to do with the dorsal pathway, the where pathway, where a sound is. So it's important how. So sound is louder in one ear than the other, so that tells you where it comes from. So we need information from both ears to locate where a sound is in space. Two main cues: interaural intensity level difference, IID or ILD, interaural intensity level, louder in one ear than the other. louder in one ear than the other. Inner oral intensity level. Inner oral time difference. So sound gets to one ear faster. So think about time. So how do we know where it comes from? It's the interoral time difference, the ITD. Think about time. Sound gets to one ear faster than the other, which tells us where it came from. Interoral intensity level difference is louder in one ear than the other. Both focus on where a sound is on the horizontal. So our pinna provides information about how vertical a sound is. The pinna is the outer part of the ear. It tells us the vertical, about how vertical a sound is. But the inner oral intensity level difference and the inner oral time difference, the IID intensity difference and the interoral intensity time difference, not intensity time difference, time difference. Intensity is the sound, time is how fast it gets to one ear from the other. These both focus on where a sound is on the horizontal. But when it comes to vertical, that's the pinna. The pinna. So inner oral intensity differences. So intensity is the loudness. The ear that is closer to the sound hears a louder sound than the ear that is farther from the sound. The difference in loudness here is a difference in intensity. Our head provides a shadow effect over the far ear based upon the difference in intensity. Our brain can calculate where the sound was. Inner oral time differences. The ear that is closer to the sound hears the sound earlier than the ear that is farther from the sound. Based upon the difference in time, our brain can calculate where in space the sound was. Difference in time, difference in intensity. So IIDs, the inner oral intensity differences and the inner oral time differences. So which is which? So difference in intensity, you get a difference in frequency, right? So here's a little diagram. So if you get a, so that'll be intensity. If it's only like one gets to one ear closer. So in a class, he was like, well, which one is which, right? It's like, which is which here? And so we have two different frequencies here and two here, but one looks more intense than the other. So this is depicting interoral intensity difference, right? So you know it's getting here faster, and this is more quiet due to this diagram of this frequency level here. And here both frequencies are the same. So this is a time difference here because one is getting here quicker than the other. So maybe you just say here this is getting quicker than the other. You could just tell by looking at this. They're both interoral, right? But which one's the intensity difference and which one is the time difference? The intensity difference would be this one because these are two frequencies, but one's different than the other, which is one's louder than the other. And the one that's more louder is getting to that ear faster. And the time difference is both the same, but one got there quicker than the other. That's how we were able to tell in this little very short diagram. And so the pinna, remember the pinna processes vertical information. So it tags different sounds, makes certain tags. So owls don't have pinnas, but they have very good sound, right? So the interoral time differences and the interoral intensity differences. Why was it ILD though? Oh, no, interoral level difference? No, we'll just say intensity difference because that's what my brain is remember. So interoral level differences is the same as interoral intensity differences. So the ITDs and the ILDs only provide information about where a sound is along the horizontal. We use our penis to locate where a sound is vertically. Head-related transfer functions. Um, Interesting note. Changing the shape of the pinna changed localization abilities. Eventually, subjects learn to localize sounds properly. Eventually, the ear learns again. The ear and the brain learn again. So you change the shape of the pinna, you change the localization abilities to know where sound came from if you change that. So we'll just do another video for smell separately. So smell. We already did vision. Somato sensation. That's skin. That'll be pain, right? What about smell? Well, that's different. So yeah, we'll do another video for smell, and then we'll do touch. So you have to look for it. So that's the somatosensory pathways. But we'll do another. What about smell? Peace, y'all.